The views expressed on Skeptically Speaking are not necessarily the views of this station, its affiliates, sponsors, or advertisers.
Bring it on, the skeptics await your letter. Your call, tweet, homing, pigeon, whatever. Desiree Schell gonna raise a little hell, metaphorically that is. Cause we're skeptically speaking.
Hello and welcome to Skeptically Speaking. I'm Desiree Schell. This week we're learning some fascinating facts about feces. We'll find out how scientists look at fossilized poo for clues about the evolution of human gut bacteria. And we'll find out how some species of dung beetles navigate using the night sky. But first, let's find out about the sometimes controversial practice of fecal transplants.
This is Skeptically Speaking, and I'm Rachelle Saunders. I'm here with Maryn McKenna, a journalist and author who writes about public health, global health, medicine, and food policy, where she's sometimes fondly referred to as the scary disease girl. She has written 2 books, Superbug, The Fatal Menace of MRSA, and Beating Back the Devil, on the front lines with disease detectives of the Epidemic Intelligence Service. She is a blogger for Wired, a columnist and contributing editor to Scientific American, and has written for a long list of other national and international magazines, including Nature, Slate, and The Guardian. Today, she's here to talk about fecal microbiota transplantations, more commonly known and much easier to say as fecal transplant, a medical procedure that's gaining quite a reputation. Hello, Maryn. It's great to have you back.
Hi, thanks for having me.
So, what is a fecal transplant?
It's just what it sounds like. A fecal transplant is where you take feces from the colon of one person and you put them into the colon of another person, with that recipient's colon having been prepared for it by being cleaned out. And this is done when the bacterial flora that live in each of our intestines become very disarranged by disease or by a chronic condition. In the United States, this has only been accepted really for severe Clostridium difficile. In other countries, particularly in Australia, it's being done for other things such as IBS, IBD. The thought behind it is that our intestines, when they're working well, represent a healthy, interacting bacterial community that's self-sustaining and that has a lot of influence on other processes in the body and on our immune systems. And when that gets disarranged, the easiest way to restore it is to kind of borrow it from somewhere else. The procedure differs a little bit depending on where you're doing it, but the basics of it are the same. What happens first is that the person who's going to get the transplant takes the kind of really intense laxatives, basically, that you would take if you were going to get a colonoscopy, where a lighted tube is slid up the colon and into the large intestine to look for any preliminary cancers. So there's a really intense set of laxatives that people take for 24 to 48 hours that make sure that everything down there is completely cleaned out. Then poop is given by the donor and is diluted in some way into something that's more liquid, and then it is placed into the recipient's colon. Now, in the United States, that's mostly been done with an endoscope, which goes in from the colon and up into the junction between the small and large intestine. In other words, it goes pretty far into the large intestine, largely so that they're sure that the liquid's going to stay there for a while and the bacteria can kind of get used to their new home. Some work in Europe has used what's called a nasogastric tube, which is a long, slender tube that reaches the colon by going in through the other end, by starting at the nose and snaking all the way down through the digestive system. There's also a sort of do-it-yourself option that appears a lot on the internet, but actually has been tested by an academic in Canada, which involves just a commoner garden enema kit, the sort of thing you'd buy at a pharmacy. People didn't think that enemas were actually going to work because an enema tube, for safety, isn't actually that long. It just goes into the extreme end of the colon, sort of closest to the outside world. People thought, oh, if you put the bacterial solution there, it just wouldn't stay and the bacteria wouldn't get up to where they really needed to be. But in a really small trial, plus all the many uncontrolled, unstudied accounts that you see on the internet, something as simple as an enema seems to work as well.
Huh. What I found interesting is even though this treatment is getting a lot of press, it's not actually that new. In some form or another, it's been around since the 50s.
The first documented use and kind of recommendation of this is in the United States in the early 1950s. I actually remember reading a book that was published, I think, in the 1960s. That's an account of someone doing their medical internship at Bellevue Hospital in New York City, which used to be really famous as sort of the toughest of the tough. The book's called The Making of a Surgeon, and it's a real kind of cowboy, tough, testosterone-laden account of what it was like to be a doctor back in those days. And he recounts that they had a patient who had severe antibiotic-associated diarrhea. You take antibiotics, they kill the bacteria in the gut, and then all kinds of bad things happen. And they cured it quite illegally and covertly by actually making essentially a fecal milkshake for a patient and feeding it to him or her. I've forgotten who the patient was. And the patient got better. And it took, years later before the guy fessed up to actually having done this.
So going back to sort of modern day use with people, the fecal transplants have unheard ofly good recovery rates.
It's really amazing. It took quite a while for medicine to start to take this seriously, in the sense that medicine wanted to study it and to see whether it was working. And the leader in this actually is a physician who's still practicing in Australia, whose name is Barodi, who has an entire clinic devoted to this. But once other researchers started to look at what he was doing and started to look at what their patients were asking for and what they, in some cases, were performing to give their patients a break, they find rates of cure, flat out cure for Clostridium difficile, which is the most studied of these conditions for which fecal transplant is used, that are way up in the 90s. 90%, 92%, 98%. If you go into patients who maybe didn't get it to quite take the first time, but have had a second round, there is no drug that works that well. And this is not a drug. It's uncomplicated to do. It's relatively safe, though there are some important caveats to that. It's also not very expensive most of the time, and it works astonishingly well.
So you mentioned safety. Now, I have heard that this, by and large, is pretty safe, but what kind of health concerns might be associated with a transplant like this?
There are a few sort of mechanical risks if you're doing this yourself. You could actually hurt yourself. If you decide to use the wrong equipment, or if you use it badly, you could actually injure your intestines in some way. But the more serious concern is that you don't choose your donor well. Possibly, since what you're doing is you're passing along a complex mix of bacteria, there could be in there disease bacteria, or the person could have some chronic condition maybe that they're not even aware of, and the recipient of the transplant could acquire that infection or chronic condition at the same time.
You're listening to Skeptically Speaking with Rachelle Saunders. We're here with Maryn McKenna, author and public health journalist, talking about fecal transplants. You mentioned screening. So how do you go about finding a donor if you're looking to get a fecal transplant? Is that the responsibility of the patient? Is it the responsibility of the doctor? Is it a combined effort?
Because there's not yet really any fully agreed upon protocol to how to do this, there are different things that people do. Now, there are some academic physicians, gastroenterologists, who have set out sort of best practices, but I don't think they all agree on this. Most people that I have spoken to will ask their patients to find someone, usually to whom they are related, and sometimes someone living in the same house. And the thinking behind that is that because this is literally a pretty intimate procedure, it's good to be asking it of someone with whom you have a relationship as opposed to a total stranger. But there are other folks who say that's actually the wrong approach, because if you've got something wrong, the chances that a person who shares your environment might also have that thing wrong, maybe without even knowing it, are not trivial. And so you should in fact go for someone who's completely unrelated. At any rate, once the donor's found, whether it's someone who is related to you or your spouse or partner or someone in your household or family or someone unrelated, they generally go through the kind of testing that you do for someone to be a blood donor. That is looking for any kind of bloodborne disease. Do they have hepatitis or something like that? Something that could potentially be passed on when a biological material is passed on. If they're clear of that, and if they report that they don't have any gut trouble, they don't have chronic diarrhea or chronic constipation, they don't themselves have irritable bowel syndrome or something like that, then the donation proceeds.
So this procedure has been performed and been getting pretty good recovery rates since 1999, but until recently, it's really only been a niche therapy. What's stopped it from catching on?
I think of it as the ick factor. And it's really interesting that gastroenterologists, who, after all, are people who have chosen as a profession to deal with the intestines and what's inside them, really find this a kind of icky thing to do. It must be said, it's also an uncompensated thing to do. There is no, or was not until very recently, any kind of diagnosis code by which you could bill for having done a fecal transplant. So the gastroenterologists who were most sympathetic to suffering patients and most thought this was worth a try would essentially be donating their time and the time of their staff, and would only be able to charge for the use of an endoscopy suite, the room in which these things happen, and the equipment at a local hospital or clinic. But it is, it's a kind of uncontrolled thing. It's not something that's been studied. It's not a substance that has been approved by regulatory authorities, which is a problem in and of itself that we can talk about. And so whether physicians have been willing to kind of enter into the space of doing this is really kind of up to their personal sense of risk and openness to new ideas.
So there was actually a trial done by a group of Dutch and Finnish researchers. Can you tell us a little bit about that one?
There have been, in the past couple of years, several academic papers written about fecal transplant, attempting to figure out if this actually worked, or if it was just kind of sort of an urban legend, or were people seeing just a sort of enhanced placebo effect. But almost all of those papers had been sort of, I guess you could say, they were case series. They were accounts of looking back of the patients that various physicians or groups of physicians had treated. And the results looked really good in those case series, but no one had done the classic randomized controlled trial going forward, where you start with people who have not yet been treated, and then you give them the treatment, and then you compare them to people who have not had treatment or have had a different treatment, and then you assess at the end how well they did. And finally, someone did that. This group whom you mentioned, their trial was published in the New England Journal of Medicine in January 2013. And it came out astonishingly well. A cure rate that was beyond what anyone expected. It was a very small trial. There were 16 transplant patients. One of them had to drop out for reasons that were unrelated to the actual transplant itself. 13 were cured on their first round. 2 more were cured on a second round. That's way up in the 90s. I think depending on how you do the math, it's a cure rate of about 94%. Their comparison group was people who were taking the last ditch drug, the antibiotic vancomycin, which is sort of the standard of care for really severe Clostridium difficile diarrhea. And the vancomycin-only group's cure rate was in the 30s, 30%, 31, 32%.
Wow.
So in the 30s compared to the 90s, you can see why people want to do this.
Yeah. So you mentioned that there's a bit of a strange roadblock in getting this approved by the FDA, and some kind of logistical problems as to who should control poop when it's kind of medicinal.
So if you think about poop, it doesn't really fall into any of the categories that the US FDA uses to study and approve things. It's not a drug. It's not a device, and it's not even technically what the FDA would call a biological, something like an implantable tissue or a lens transplant or something like that. So the FDA has had a lot of difficulty in figuring out how to set out the rules by which academic researchers could approach the FDA and begin a trial. In order to do that, in order to begin a trial, the FDA has to have sort of acknowledged that you're going to do it and engaged in negotiations with you. And finally, they did agree to do that. This is going back almost a year now, that a group of physicians who have been the most sort of public and forward about the need for fecal transplants got the FDA to agree to the structure of a trial. And that trial is actually ongoing in several locations, including in Rhode Island and in New York. In the meantime, though, sort of the buzz about fecal transplants got louder and louder, and there was no way to say, are people doing it well or badly, or are people doing it themselves? And the FDA started to get concerned. And so back in May, they imposed a requirement on any physician who wanted to do a fecal transplant, saying that it would have to be done under what's called an investigational new drug application, or an IND. Now, an IND is not a trivial thing. It requires a lot of paperwork. It requires usually about 30 days of review to ask the FDA if you can use your investigational new drug, which in this case would be poop, and you're not guaranteed a yes. And the FDA said, for anyone who wants to do fecal transplants for patients, we're going to require an IND. And there was an immediate uproar. And a number of academic researchers and private physicians as well just kind of poured onto the internet and said, this is just completely inappropriate. The risks of this procedure are not sufficient to justify the amount of hurdles that are being put in front of it. And the FDA, to their credit, thought about what this outcry was presenting to the agency and actually backed down. And about a month later, sort of moderated its tone, not to the sense of saying you can return to the Wild West, but they did indicate that an IND would not be necessary for every patient. However, they wanted to be sure that physicians were going to be doing a good job of making sure that patients fully understood the procedure and all its risks, of which, as we talked about, there are a few.
So I read a little bit that right now we're using fecal transplants as sort of the last resort, but there's quite a big push actually to have this be the first resort, in part because a lot of the medicines we're using to try and treat this don't work very well, and we're running out of them.
Vancomycin is not a benign drug. It is, one of the toughest broad spectrum big antibiotics that you use kind of when you've run out of everything else, when things are extremely resistant. It's not used only for C. diff, to use the sort of affectionate name for Clostridium difficile. It's also used for highly resistant infections like MRSA. And after vancomycin, there isn't a lot out there. So medicine as a whole has an interest in keeping vancomycin from being overused. In addition, vancomycin is not an easy drug to take. I mean, one of the reasons why it's lasted so long on the market, it's been around for more than 50 years, is that for many years, it was considered so sort of harsh a drug that it kind of stayed on the shelf while people used other things. So people who take vancomycin generally don't enjoy it, though they generally are sick enough that they really, really need it. So since it's not good to use the drug if we don't need to, and since it's not a good drug for patients to take unless they absolutely have to, and since there is another treatment, fecal transplant, that we know works really well, really rapidly, a number of physicians who've been studying this and using this procedure have actually come forward in a medical journal and said, we really think that this should be the standard of care, that it shouldn't be the thing you do after you've tried all the drugs and they haven't worked and the C. diff has come back. We should do it right at the start.
This is Skeptically Speaking. We're discussing the trials and tribulations surrounding fecal transplants, including their effectiveness and the roadblocks to getting them, with science journalist and blogger Maryn McKenna. In January, you wrote in a blog post on Wired that I have heard from more than 100 people who are afflicted with C. diff, believe that a transplant could help them, but cannot find a doctor who agrees that the procedure has merit. It sounds like most of the resistance historically has come actually from the medical community and not the patients who are afflicted.
Patients are just desperate to find people to do this. They are much more open to it than physicians have been. And, I don't want to suggest that physicians are corrupt or stupid or just looking for money. I think they've just been being very cautious because this is still an unapproved and somewhat underground procedure. And, we live in a very litigious society. I think as a result, they're very risk averse, and they're looking for some sort of regulatory blessing on this, which is why the FDA's involvement is going to turn out to be so important. And also because everyone finds or almost everyone finds their own donor, the physicians are kind of locked into using material that they don't really know very much about, because the donor is coming to them fresh, even as the patient is. Now, there's a couple of places that are trying to work around that. As I mentioned, there are a few medical schools and practices that have identified kind of perfect donors, people whom they ask to donate repeatedly. At the University of Minnesota, they have been working with they have a pool of donors, and they kind of put together their perfect transplant, and then they actually freeze it, and they keep it in their lab freezers and thaw it out as necessary. So they've kind of established a donor pool. The thing that everyone looks forward to, in some cases with anticipation, in some cases with a lot of dread, is there going to be at some point a reliable, artificially produced transplant material, a sort of fake poop? That would be good on the one hand, because it could be, you wouldn't need a donor. You wouldn't need to find the right person, and you'd sort of eliminate the ick factor, because you wouldn't actually be dealing with feces anymore, but just with kind of the bacterial component. But on the other hand, at the moment, feces are really abundant and essentially free. And once you turn them into a product of the pharmaceutical or medical device industry, they may turn out to be equally abundant, but they're certainly not going to be cheap anymore.
Yeah, I feel like having this become a synthetic product really opens the door to problematic patents and costs that really change the landscape for something that has been, one of its sort of selling factors is that it is, a renewable resource.
Exactly.
And requires very little effort to produce.
Now, there is a team at the University of Guelph who have been working on a fake poop. They actually have a kind of digester that I think they call RoboGut. So that is brewing a sort of bacterial extract. They themselves have said that they are not able to get the diversity of bacteria in their extract that you would get in an actual fecal sample. But they've tried it on a few people, and those few patients have recovered. Now, they also aren't charging potential pharmaceutical prices for it precisely because it's just an experimental thing. So, on the one hand, that's really promising. On the other hand, the question of monetizing it, I think, is very troubling. But on the other hand, the only way these days that you get really large trials of any new pharmaceutical or medical device is if there's some intellectual property at the end. That's what causes large corporations or even small startups to be interested, is that they're going to realize some income at the far end of the process. Until they think that kind of income is forthcoming, they're not going to help stand up really large trials. But it's the really large trials that provide the results that regulatory authorities want to see. So it may be inevitable that pharma companies are going to get involved in this process, It may make this procedure blessed by authorities. It may make it safer. It may make it more predictable, but it's definitely going to change the DIY and cheap, if not free, culture of fecal transplants for good when it does.
So beyond C. diff, what other diseases of the gut might fecal transplants actually be helpful for?
So the folks in Australia have done this for, if I'm remembering correctly, for other disorders of the bowel, for irritable bowel syndrome and Crohn's disease, inflammatory conditions of the gut. Beyond that, you get into some really interesting territory. If the makeup of the bacterial flora in your colon determines what your nutrient uptake is, and therefore essentially what your metabolism is, and whether you tend to be fat or tend to be thin. If you're someone who is obese or someone who puts on weight easily, should you seek out a fecal transplant from someone who's naturally thin? These are the sort of questions that people are asking. There's also, since the gut also forms a kind of second immune system, can we enhance the immune system by fecal transplants? Can they affect mood and mood disorders? These sound like really out there suggestions, but there are people who are starting to think about these as well.
The microflora of the gut is just fascinating, and the more that comes out about it, the more interested in it I am. Maryn, thank you so much for being here.
Thank you for having me.
And thank you for agreeing to talk about poop. If you're interested in learning more about Maryn McKenna, reading her work, or learning more about fecal transplants, you can visit skepticallyspeaking.com, where you'll find links to get you started in our show notes.
Skeptically Speaking is rebroadcast on a number of fine radio stations. To find out where Skeptically Speaking airs near you, to listen to past episodes, or download the weekly podcast, check out our website at skepticallyspeaking.com. While you're there, follow the links to connect with us on our Facebook page, our Twitter feed, or to subscribe to the show in iTunes. And now, back to the show.
Welcome back to Skeptically Speaking. This is Desiree Schell here with Cecil Lewis, Associate Professor in the Department of Anthropology at the University of Oklahoma. Cecil was the corresponding author of a research article published in the Public Library of Science, PLOS ONE, about the evolution of human intestinal flora via analyzing ancient feces. Good to have you here, Cecil.
Well, thank you very much. Glad to be here.
So tell me a bit about the research.
So what we're looking at is human microbiomes. So a microbiome is really, if you're taking an environmental sample, any environmental sample, that sample could be from your tabletop, or it could be from your body site. It could be from your skin, your nose, or your gut from fecal material. And you look at that environmental sample by its microbes, all the microbes that are inside that environmental sample. And you often look at those microbes through the lens of genomics or genetics. And that would be a microbiome. What we wanted to do is understand how the human microbiome has changed over time. Now, right now, there's a great deal of research that's being done on human microbiomes, often in clinical settings, and people that are in urban environments, basically the people around today. And we're learning a great deal about those microbiomes and how they affect health. But what we don't really know is what those microbiomes look like prior to modern practices, prior to chlorinated water or antibiotics. And so what we wanted to do is get a better understanding of the ancestral state of the human microbiome, and using one of our approaches, the one that was published in the paper that you've referenced, is looking at that ancestral state through archaeological biological material, things called coprolites. Coprolites are archaeological fecal material.
And so where did you take those samples from?
The first study we did was in 2008, and then we had the most recent study in 2012. And the study that was the most fruitful, or the sample that was the most fruitful, came from Mexico at a site called Rio Zape. And it is a cave site. It dates to about 1,300 years ago. And it's a really sort of an extraordinary cave site. There was a human occupation at that site. But then, fortunately for us, that previous human occupation was covered in an adobe layer, basically a clay-like caked layer. that preserved a great deal of that archaeological site. It basically created a time capsule, and the preservation of that material was quite good. And so those are the coprolites we study that were underneath that adobe layer. Now, there are other studies. In a 2012 study, we also compared it to previous data that was generated from coprolites from permafrost samples. And those were really great as well. But, permafrost samples are pretty rare. So being able to get this from a cave site that was not a permafrost sample and really get human gut microbiome data, that was really exciting.
Well, and how exactly did you analyze all of these samples?
Well, this particular study, we analyzed it by first targeting a gene that's found in bacteria called the 16S RNA gene. And we targeted a particular area of that gene that's known for being hypervariable. So there's lots of variation at that particular part of the gene. Now, why that's good is that the gene is not only useful for looking at bacteria in general and determining whether or not you have bacteria, but because it's really variable, it's variable among different types of bacteria, different genera or different species of bacteria. And as a result, you can get a fingerprint for the types of bacteria that are found in the gut. You basically get frequencies of the different genera or frequencies of the different species by targeting that particular gene.
How do you know that all the samples are human coprolites?
We know that it's human for a number of different reasons, but probably one of the best ways of demonstrating it was human is not only the archaeological context, but we identified the mitochondrial haplogroup of the human host themselves. So the mitochondria is this little cellular organelle, this little cellular machine that's inside of all of our cells. It's inside of all eukaryotic cells. They're the major energy producers for cells. And what's fascinating about mitochondria is they have their own circular DNA. In fact, it's a DNA that looks a lot like a bacteria, even though it's a part of our cell. And there's some evolutionary explanations as to why it might look that way. But what's important is we were able to amplify that mitochondrial region as well for the coprolites that reflect the human host and identify the mitochondrial haplogroup, which tells us a little bit about their maternal line ancestry. And what we found is for each of the coprolites, we could identify that haplogroup as a Native American haplogroup, and we actually found that there was at least two individuals represented, two different Native American haplogroups. So we could identify the human Native American haplogroup from the coprolites themselves as one line of evidence that it was a human coprolite. Among others, the archaeological context is also quite clear. The cave site can only be accessed by hand and foot holds. It would be very difficult for most mammals, for example, to access the site without human intervention. And so there's lots of different complementary data that would support that these coprolites were in fact human coprolites.
And so then you compared these samples, and what did you find?
Well, one of the things we found in our earlier study in 2008 is that the information about human gut microbiomes is there. What was important about the 2012 study is even though we know some information about the human gut is there, can we get around the things that can confound what we want to say? For example, can we get around the fact that there's going to be some level of soil penetration? And what we found is that we can for some coprolites. There are coprolites where the human gut component can be isolated through a bioinformatic method called source tracking, and it works pretty well. And after we figured out the component of that microbiome that looks like human gut, we started to learn a few things about what that human gut was like back then. In fact, what we found is that the human gut resembled rural populations that we have today, populations that live relatively traditional lifestyles. So we also found the presence of spirochetes in the human gut, which is a type of bacteria known for being able to metabolize very complex carbohydrates, cellulose-like material. And those spirochetes were also observed in rural traditional peoples alive today. And so that was kind of exciting. So these spirochetes seem to be a part of what we might call the traditional or maybe ancestral microbiome And they're not common in an urban population. They're not common in a population living a largely westernized lifestyle. So this may be an aspect of the rural core, the traditional core microbiome that has since been lost by our modern practices. And now, is there a consequence to that remains to be seen. There's a lot to think about there, a lot of hypotheses to test. But what's fascinating is that previously there was only 2 other continents really explored for traditional microbiomes on living populations, Africa and South America. So now we're seeing some of the things that we learned from Africa and South America in a sample in North America, in Mexico specifically, southern North America, that dated to 1,300 years ago is sort of another confirmation that this really does look like something that reflects the ancestral microbiome.
This is Skeptically Speaking, and Cecil Lewis is explaining some recent research into ancient poop. So I believe that the research also argues that modern use of antibiotics has affected the health and the content of our gut bacteria. Is that correct?
Yeah, so what we know is, whether it's antibiotics specifically or modern practices, it seems quite obvious that our relationship with microbes has fundamentally changed. Now, I'm not saying it's necessarily all good or all bad. There's a lot of really nasty microbes out there, and by practicing sanitation and aseptic practices and chlorinating your water, I think overall we're very happy to be doing that, given some of those little nasty critters that are out there. But our approach to removing those really nasty critters was not targeted. It was very much a shotgun approach. And there has been consequences to this, and we're learning about those consequences. We're learning that the consequences to this more sterile environment is the developmental issues in our immune system, that we're having a less developed immune system, including the emergence of more things such as allergies and similar problems. So I'm not saying that we need to stop our aseptic practices, certainly not, but I am saying that there are consequences to that. And I, in the best of both worlds, maybe we can find a way to recoup what might have been lost and find out what were the consequences in a more sophisticated way, and then potentially fixing that problem. So we can still avoid those really nasty pathogens that are out there without the side effect of maybe this increase in allergies. Maybe there are strategies that we could use to regain what we have lost, the good microbes, the friendly guys, or maybe it's not specific microbes, maybe it's microbial frequencies, regain those frequencies that can be beneficial to us.
But humans have adapted to other changes in our diet. We've built up a tolerance to lactose, for example. So why wouldn't our intestinal bacteria adapt to antibiotics?
Well, I think that we have to rethink about what we're saying here. For one, only a small subset of humanity has adapted to lactose. And when we talk about something like natural selection, for example, to take a quick step, and that's what we're referring to when it comes to the human genome, it's not without consequences. The very definition of natural selection involves some people being more successful than others. In other words, there's real life consequences to a population that's undergoing serious natural selection, consequences that we today would prefer not to happen. Now, microbes very well may adapt to some of these antibiotics that we're doing, and maybe we can come up with strategies to combat that, because we want to make sure that the nasty microbes are not the ones that are doing the adapting. We want the good microbes to be there, but the nasty microbes to stay away. And so, getting back to the intestinal bacteria, they very well might adapt to our antibiotics, and they may be developing antibiotic resistance. But we don't want that to proceed in a shotgun way either. We want to make sure that if they are adapting to antibiotics, it's not the nasty guys, which inevitably it will be. So one of the things that is also important for characterizing these ancestral microbiomes is potentially better understanding how this antibiotic resistance even happens. And then by understanding how the antibiotic resistance happens, how does it evolve? How does it come about? Has it been something that's been around in nature forever, or is it something that's really involving a lot of mutation, like novel mutation? And we don't really have a good understanding of that process very well. But importantly, we don't want that antibiotic resistance to emerge in nature in a way that we can't control, because some of those bacteria that we don't want are going to be revisiting us very, very soon.
So now again, just to reiterate, does the research support this idea, or is it more giving us a direction to go with further research?
So the research really just focuses on characterizing the ancestral state of the human microbiome, which has fundamentally changed compared to what we see in urban populations living the largely westernized lifestyle. It doesn't dive into why it's changed specifically. That why it's changed is stuff that are hypotheses to further evaluate, but pretty compelling hypotheses. Obviously, chlorinated water, antibiotic use. Maybe there's the global food network that we have now. There's a series of hypotheses that are somewhat intuitive, but need to be further evaluated as to why this gut microbiome today in these westernized populations or in these urban populations look really different. To be honest, we have a long ways to go. Most of our understanding of human microbiomes today, even though that we've been studying these using next generation sequencing for over 5 years, almost all of our understanding has come from these urban, affluent, westernized individuals. And so we really don't know what to expect still in our own backyard. There may be a lot of microbiome diversity in our own backyard that is just gone uncharacterized.
Well, that makes me think about what else this methodology might be able to tell us. A lot of traditional diets are primarily based on roots, and in other regions, grains. So that would show up as a difference?
Potentially. I mean, we really need to better understand the metabolites that are a consequence of these bacteria metabolizing what we give them. Basically, we give them food to digest, and we benefit from their metabolism. They produce products for us that we couldn't digest on our own, including vitamins, and they help us clear out cholesterol. They do lots of very important roles for us. So what it really boils down to is not necessarily what the macro ingredient looked like, whether or not it was wheat or whether or not it was a potato, but what it breaks down into, what kind of complex starches and cellulose and stuff at that level. At least as far as we understand at this time. Again, we have a lot to learn, but I think what it's going to really boil down to is what are the component parts of that food. And we might find some things that are somewhat universal. If it's a very complex starch, we might see similar trends. If it's heavy protein, we will probably see similar trends. We're already beginning to see that. But it might not be exactly the specific food, whether it's a type of potato or whether or not it's a type of wheat. But that, remains to be seen.
Well, I guess the reason I ask is that there are papers that propose that the whole ape-human split involved a root-based diet, and most tropical foragers have a root-based diet, but later agricultural people and temperate farmers, they have mainly a grain-based diet, from what I understand.
Yeah, absolutely. I think those periods of domestication will be fascinating to characterize in the future.
And they might stop a lot of anthropologist fights.
There's always going to be something to fight about, right? But I think we're going to learn a lot about what these transitions, these different moments of domestication, we potentially have the exciting opportunity to learn a lot about how it has impacted our microbial self. And, I think one of the important messages here, too, is our microbial self is a substantial self. I mean, it's a huge part of us. And I think that's one thing we haven't quite talked about yet. But one way of looking at that might seem small until you really think about it is are the microbes that are on your body, for example, is about 2% of your weight. If I'm gonna round up here, all right, I'm rounding up. If I'm 200 pounds, that means that there's roughly around 4 pounds of microbes on me. Now, 4 pounds for a 200-pound person might not seem like a lot, but your liver weighs 3 pounds. So that's a pretty sizable organ, really. But there's other ways of thinking about the number of microbes that you carry. So the human body is composed of roughly 100 trillion cells. But the microbes on the human body is 10 times that number. There's 10 times the number of bacteria cells on the human body than there are essentially human cells. So your body is essentially 90% bacteria and 10% human if we use the cell as the unit of analysis. And that's an extraordinarily high number. I mean, 10 times that number, that's 1 quadrillion bacteria on the human body. 1 quadrillion. And it's hard to get your head around that number, but there's 300 billion stars in the Milky Way galaxy, roughly. So it would take 3,000 Milky Way galaxies for the number of stars to equal the number of bacteria on a single human body.
Thanks for putting it in perspective, sir. And Cecil, thank you so much for being here.
Well, I appreciate it. It's been fun.
You can find a link to Cecil Lewis and the +1 article that we've been talking about on our website at skepticallyspeaking.com. And we'll be right back with more of this very special poop episode after this.
Next week, Skeptically Speaking becomes Science for the People. Hosts Desiree Schell, Rachelle Saunders, and Marie-Claire Shanahan discuss the new name, the motivation for the change, and how it conveys their vision for the show. We'll speak to researcher Alice Bell about the history and politics of the radical science movement of the 1970s. And researcher and blogger Cy Curious gives us a preview of Girl Geek Con 2013. That's next week on Science for the People, on your local radio station, or online at scienceforthepeople.ca.
Welcome back to Skeptically Speaking with Rachelle Saunders. I'm here now with Dr. Eric Warrant, professor of zoology and director of postgraduate studies in biology at the University of Lund in Sweden. Professor Warrant leads a research group studying vision in nocturnal and deep-sea creatures. Eric has also collaborated with Toyota to develop night vision systems for cars, and is part owner of the company Nocturnal Vision AB. The study we're talking to him about today, how dung beetles navigate using the Milky Way, recently won a joint Biology and Astronomy 2013 Ig Nobel Award from the Improbable Research Organization, and you can learn more about them by visiting their website at improbable.com. Congratulations and welcome to the show, Eric.
Thank you very much. I'm very glad to be here.
Okay, so before we jump into your study specifically, let's get everyone up to speed on our little friend, the dung beetle.
Dung beetles are amazing animals, among my favorites, I have to say. Dung beetles live, as their name suggests, on dung, and they eat dung. They breed in dung. And the beetles that we've been working mostly with in South Africa actually roll dung. There are 2 types of dung beetles, those that roll dung and those that just simply dig it down below the pile of dung that's left by an animal. But the beetles that we've been working on roll dung, which means that they create a ball and then they roll it away from the dung pile and bury it. And they also lure to themselves a female. It's usually the male that rolls. They lure themselves a female using often pheromones. And then the female and the male mate, and the female then lays her egg in the center of the ball, and then this baby beetle develops inside the ball, which has then also been buried under the ground by the pair.
So is this dung ball rolling a nightly occurrence, or is this something that kind of happens seasonally?
Dung beetles are mostly active during the warmer months of the year, so spring, summer, autumn is their major season. And dung beetles tend to be either day active or night active. In other words, there are nocturnal species and diurnal species, and there are other species also that are active at specific times during the dusk and dawn. So they've really picked specialized times to be active in order mostly not to compete with one another. And if the species are active at the same time, then they're more likely to find males and females of the same species to mate with at that same time.
Now, I've heard that it is important for the dung beetle to roll their ball of dung in a straight line, sort of away from the pile. Is this true?
It is indeed. And the reason they have to do this is that the dung pile itself tends to be a ferociously competitive place, especially in places like Africa where dung is in short supply, but dung beetles are rather numerous. So once a dung pad has been dropped by an animal, it's not very long at all before many, many beetles are inside that dung pad, and they're all rolling balls as fast as they can. And the thing is that just as in the human world, in the dung beetle world, there are big, tough, lazy males hanging around to beat up anybody who's made a nice ball and steal the ball off them. So in order to reduce the chance of this happening, once you've made a ball as a dung beetle, you've got to get away from there as fast as you possibly can. And the most efficient and safest way to do that is in an exact straight line.
So how far away would the average dung beetle roll his prized ball of dung?
That depends very much on the species, but typical distances could easily be in the range of 20 to 30 meters, or
Wow.
shorter, or sometimes longer.
Wow, that's impressive for what I'm assuming is a fairly small insect. What prompted you to look into how these dung beetles navigate?
Mostly it was the observation that they roll in straight lines, which is not a trivial thing, actually, especially if you consider how many body lengths a typical dung beetle rolls over. I mentioned before you could easily roll 20 or 30 meters as a dung beetle. And if you're a small animal that's only a couple of centimeters long, that represents quite a long distance, maybe kilometers even in human terms. And so we started to wonder how on earth they managed to achieve this. And it was known before that some insects are able to walk in straight line paths by looking at a pattern of light in the sky that we can't see. And this pattern of light is actually made up of polarized light rays, which are produced by scattering of sunlight from particles in the atmosphere. And this pattern is very symmetric and very obvious to animals that can see this type of light.
So this polarized light, how do they see it? What gives them that ability?
The ability comes from having photoreceptors that are built in a very special way. The photoreceptors are the light-sensitive cells in the eye, and the way they're constructed makes them intrinsically sensitive to the direction of polarized light in the sky in a way that our photoreceptor construction does not allow. Invertebrate animals, that is to say animals without backbone, tend by and large to have the kind of photoreceptors that are built with the intrinsic ability to detect and analyze polarized light, whereas the photoreceptors of vertebrate animals like ourselves are not.
So tell me a little bit about your experiment setup. I understand you did both outdoor and indoor experiments, correct?
That's right. Actually, we do all sorts of different kinds of experiments. The outdoor experiments and indoor experiments that we did in South Africa, for instance, were completely behavioral. That is to say, we observed beetles rolling their balls of dung and manipulated celestial cues in the sky to see what effect that manipulation had on their rolling directions. And those experiments we've been able to perform outside under a natural sky, and we've also performed these experiments under artificial conditions in a planetarium.
I also read you put at one point little cardboard hats on your test beetles to block their view of the sky. That sounds adorable.
Yep, that's right. We constructed little cardboard hats which we taped onto their heads, which indeed prevented them from seeing the sky. And the reason we did this is that we wanted to see what would happen if these cues, like the sun's disk itself, in the case of a day-active beetle, or the polarized light pattern in the daytime sky, if these cues were blocked, what effect that would have on their straight line orientation.
You're listening to Skeptically Speaking with Rachelle Saunders. I'm here with Dr. Eric Warrant, professor of zoology at the University of Lund, and we're talking about dung beetles and how they navigate. So what exactly did you guys find out after all these experiments?
Well, the interesting thing was that if you take a day-active beetle and put a little cardboard hat on their head, they're completely incapable of rolling their balls of dung in a straight line. In fact, they just go around and around and around and around in circles. As soon as you remove the hat, they're fine, and once they can see the sky again. And we've found that there are basically two important cues in the sky which are used. The polarized light pattern I've just mentioned is one of them, and is probably the most important actually of the two. And the other is the disk of the sun itself. And even we are able to use the sun as a kind of a directional cue to find our way.
So that's during the day. What happens at night when the sun isn't around?
Now, that's a very interesting question because the night sky is compared to the daytime sky quite complicated. You can have a similar situation at night as you have in the day. You can have a heavenly body at night, that's the moon, and around the moon, just as around the sun, there is a pattern of polarized light produced by, in this case, the scattering of moonlight by particles in the atmosphere. And just as during the day, this pattern acts as an excellent compass cue for straight line orientation. So we actually find that on moonlit nights, nocturnal dung beetles also roll their dung balls in straight lines over considerable distances, and that this rolling behavior is totally disrupted if we put a little cardboard hat on top of their heads to block out the view of the night sky.
Now, my understanding was that somehow they were also using the Milky Way to navigate. Is that correct?
That's right. That was quite a shocking discovery, actually. One evening when we were in South Africa, we're always there long enough to go through periods of the month when the moon takes a long time to come up. And for a very large part of the night, there's just a very starry evening. And we saw on one evening that the beetles that we were doing experiments on still rolled their balls of dung in a straight line. And we started to get very worried when we saw this, because we suddenly thought, well, maybe everything that we thought we discovered earlier is just simply not true, and they're using some other cue. And then it dawned on us that maybe they could use the stars as compass cue. But the problem there was that previously people had thought about these kinds of issues in other types of insects and had come to the conclusion that insect eyes are neither sensitive enough nor have sufficient spatial resolution to actually see very many stars in the sky. But the interesting thing about the southern hemisphere night sky is that the Milky Way is a very prominent stripe of light in the sky, which we don't see quite as clearly in the northern hemisphere. And our hunch at that point was, well, maybe these beetles are able to see this broad stripe of light in the night sky in the southern hemisphere and actually orient relative to that if the moon and its polarization pattern are missing.
So you guys actually sort of replicated this in an interior setting so that you could control everything. Can you tell us a little bit about those?
We did a number of experiments out in the field first, where we created an arena where it was possible for the beetles only to view the night sky and nothing at all other than that. And then we could test whether or not they could roll in straight lines with their dung balls, and indeed they could. And so we decided at that point then to move the entire apparatus into the Johannesburg Planetarium to repeat the experiments, but this time under a night sky that we could manipulate. We were able to turn on and off various groups of stars in the night sky. It was fantastic, actually. So we could, for instance, leave all the stars on in the natural way, or we could remove the 18 brightest stars and leave all the dimmer stars, including the broad stripe of light of the Milky Way. Or we could remove all stars altogether except for the Milky Way. And then, of course, we could turn all the stars off totally and have it completely dark. And when we did this experiment, we discovered that the stripe of light that represents the Milky Way in the planetarium was sufficient on its own to allow them to roll in straight lines.
So how unique is this navigational tactic among, I guess, the insect population of the world and I guess the animal populations as well?
So far, at least, this is the only animal that we know of that uses the Milky Way for orientation at night. In theory, humans could do the same thing if we kept looking upwards, and maybe some people have done this, but at least in the animal kingdom, no report of this has ever been made before. Now, that by no means, of course, that the dung beetle is the only animal that does this. There are plenty of nocturnally active animals like birds and many other kinds of insects, other beetles, moths, that are all capable nocturnal navigators. And it's certainly not impossible that they too are able to use the Milky Way in the same way.
So what would happen in more urbanized areas where there's a lot of light pollution that the dung beetles also have to kind of deal with?
This is a problem that's more an issue in the northern hemisphere, where the population density is a lot greater. Most of the places where you find dung beetles in the southern hemisphere, in Africa in particularly, are reasonably light pollution free. But of course, even in Europe, we do have dung beetles, and we even in southern Europe have ball rolling dung beetles. And this must be a real issue for these animals, actually. Light pollution is an increasingly serious issue. And animals that are nocturnally active and use their visual systems for orientation and navigation, and indeed just for the general activities of life itself, have to deal with light pollution. And light pollution also, sadly, not only is its intensity very different compared to the natural night sky, but its color is also very different. It's much, much redder than natural light.
So the different color of light, would it confuse sort of directionally? I'm not sure what the color of light would mean for an animal.
Well, for a dung beetle, it probably wouldn't mean terribly much. But for a nocturnal flower visiting moth, on the other hand, that has color vision in order to see the colors of flowers at night, a very odd illumination spectrum due to man-made sources could disrupt their color vision system and actually decrease their ability to find and feed from flowers. And that actually is quite serious.
That is fascinating, sir. Eric, thanks so much for sharing your research with us today.
You're very welcome.
If you want to learn more about Professor Eric Warrant or his research, you can visit skepticallyspeaking.com, where you'll find a link to his website in our show notes. And while you're there, we'd love it if you'd click on the links to our Twitter feed, our Facebook page, and our Google Plus feed, where you can get the latest news from the Skeptically Speaking team and chat about the show in the comments. You can also seek us out on iTunes, where you can subscribe to the show and access all our past episodes for your listening pleasure. Thanks for listening to Skeptically Speaking.